Hi, I'm Morgan Gunning from the Blood Transfusion Service of Zurich in Switzerland, and today I'm excited to share our journey in unlocking the entire blood group genome through adaptive sampling. Currently, we have unraveled 45 blood group systems, coded by 50 genes and accounting for over 350 antigens. Accurately identifying blood groups and ensuring donor-recipient compatibility is crucial, especially for patients receiving regular transfusions like those with thalassemia or sickle cell disease. Indeed, repeated transfusions can lead to the development of antibodies against non-matching blood groups, making the finding of compatible donors challenging. Blood groups are routinely determined in blood transfusion centers through a variety of molecular and/or serological tools like the one listed here. While these methods are effective in determining most blood groups, they have their limitations, especially when SNVs need to be phased to the maternal or paternal allele or when dealing with hybrid alleles. Hybrid alleles are frequently arising in two systems, the Rh and the MNS system. Each system comprises two highly homologous genes situated in close proximity. Therefore, rearrangement between these genes are common and can highly complicate their typing. And this is where adaptive sampling comes in place. In our study, we focus on approximately 8.6 megabases of the human genome, including blood group, transcription factors, platelet, and neutrophil antigen coding genes. Reads were mapped against the T2T genome, rebase called with a super accuracy model implemented in the RADO, and for validation purposes, we compared variant calls obtained by Claire with MALDI-TOF pre-type data for up to 17 blood group systems. We've sequenced until now 4 different samples, each processed on individual PromethION Fow Cells. Despite employing the same protocol, we observe variation in output between runs, ranging from 37 to 80 gigabases. As you can see, the latest version of MinKnown exhibited enhanced performance. For those unfamiliar with adaptive sampling, rejected off-target reads are highlighted in purple on the graph below that I copied over from MinKnown. And while zooming over the first 1,000 base pairs, you can see that most ejected / off-target reads are around 650 base pairs. By allocating one PromethION Fow Cell per sample, we achieved coverage ranging from 38 to 83x across target regions, compared to approximately 9 to 22x outside the target regions, which resulted in roughly 4x enrichment. The MALDI-TOF data showed 100% congruency with the nanopore data. However, resolving hybrid alleles in the Rh and MNS system for sample 1 and 2 proved challenging with tools like Sniffle2 and required more advanced analyses beyond the reference-based method. For sample number 3, we successfully phased novel variants in the RHCE gene of a young sickle cell disease patient and thereby enhancing the transfusion guidelines. Finally, for sample number 4, our sequencing data revealed a novel 8.6 kb deletion in the RHCE gene, thereby elucidating a long-lasting, unexplained RHCE genotype-phenotype discrepancy. In conclusion, adaptive sampling emerges as an effective and time-efficient tool for characterizing the entire blood group genome. While it has successfully uncovered a novel large deletion, addressing rearrangements between homologous regions remains a bioinformatic challenge. Nonetheless, adaptive sampling demonstrates significant potential to unravel the individual blood group genome, thereby potentially enhancing patients' outcomes, particularly for those who require chronic transfusions. Before I conclude, I would like to express my gratitude to our funding sources, collaborators, and all those who contributed to this study. Thank you for your attention.